Hey guys, we're going to go through DNA replication, and first step is helicase unwinds and unzip the DNA molecule. So if you guys have a look over here, this is our twisted ladder or our double helix. So double helix. Now with the help of helicase, it unwinds. So unwinds here. And it also unzip the DNA molecule. Okay, so these are all done by the enzyme called helicase. What I'm going to do here is I'm going to write unwind by helicase and unzip by helicase as well. Now, so what does a helicase do? Helicase breaking apart the H bond which hold the bases together. Now, if you guys have a look here, we have our unwinded DNA. We have one strand over here and another strand here. Now, what does the helicase do? It breaks away the H bond that is connected in between the base. Okay, the H bond between the base. Now, you don't really need to know how many H bonds there are for between adenine, thymine, or cytosine, or guanine. All right, you just need to know it breaks the H bonds. Okay, so keep it simple. Now this creates replication fork. So what does it mean by replication fork? Here we have a replication fork. Fork. Cool. So it kind of looks like a fork. So going through it again, helicase, first of all, unwinds the double helix DNA. Now, it breaks away the hydrogen bond between the base, and this is the unzipping part, right? And it creates something called the replication fork. So if you guys look over here, this is the replication fork. You can think the helicase as a zipper. So it unzip, and as you can see, the DNA breaks apart. Now, this does not just occur in one part of the DNA. The replication fork can occur in multiple places in a DNA molecule. Okay, so DNA replication occurs in multiple parts, not just starting from one bit and work its way through. It starts in multiple parts. Okay. Now, the two parent strands of DNA are anti-parallel. What does that mean? They run in an opposite directions. Okay. This part is the complicated part, and some of you guys don't understand it. We're just going to do a very simple way to talk about leading and lagging strands, okay? Now, this is a very good little image to show you guys what does it mean by leading and lagging strands. For example, over here we have a leading strand. It goes from 5 carbon to the 3 carbons, okay? How do we know that? Oh, and by the way, when we write a line this way, we draw an arrow like this. How do we know this way? We know it by reading this frame, right? If we read the bases, it's ATCGG, CTA, ATCG, TGCT. Okay, just imagine you're reading it on the slide away. You can read it really easily. And then for the 5 carbons to the 3 carbon, we call this the leading strand. Now, the other strand is the opposite direction. It is going this way, and it is going from the 3 carbon to 5 carbon, but in the opposite direction. Now, for this particular strand, we call the lagging strand, and based on the base position, you can see it's the reverse, right? So AAG, CA, CGA, TT going this way. So we're going to just draw it over here, going this way, and the 5 strand is going this way. All right? So there will always be anti-parallel. One strand is going down this way, and the other strand is going down this way, okay? So this is the first step of DNA replication. Okay, guys, now we are going to step 2. So the parent strands have been separated with the help of helicase, okay? Sometimes we can draw the helicase like this to show that it actually separates the DNA replication. So I'm going to draw a big circle here, and we're just going to label this helicase. And if you guys are just wondering, what I did is instead of showing the DNA replication going this way, I made it into this way, okay? So now, the blue one still represents the leading strain. We can read it really easily because we can read the bases downwards, okay? So this is always going from 5 carbons to the 3 carbons. Now, the lagging strain is going the other direction, okay? Still 5 to 3, but it's the other direction, okay? So last time I wrote 3 to 5, that is also okay. But as you guys can see, we cannot read this really easily because it's going the other directions. So there is a difference between leading strands and lagging strands. You don't need to necessarily know about the positions of the carbon, but you do need to know about like the 5 and the 3, okay? You don't have to say it's the 5th carbon or the 3rd carbon. Just say it's going from the 5 direction to the 3 directions, okay? So DNA is always anti-parallel. One strand is going downwards in a 5 to 3 direction. We call this the leading strand because it is really easy to read. The other one is the lagging strand, is going again 5 to 3 but the other direction. That's why it's called anti-parallel, and it is going the other way, so it is much harder for us to see. Now in step 2, the separated parent strands are paired up with new nucleotides. Okay, where do we get these new nucleotides? We get them from foods and nutrients. Okay, and now this process is with the help of DNA polymerase. This is the second enzyme that we're going to talk about in DNA replication. First one is the helicase. So again, this is enzyme. Now we are going to talk about DNA polymerase. Now DNA polymerase basically joins the new nucleotide together and binds them with the parent strands. Okay, so we're just going to go over here and we are going to draw the DNA polymerase. Let's just use red. I like red. Okay, so just a blob here, and we're going to call this DNA polymerase. So what is going to happen? Well, we get new nucleotides. For example, we need A. Okay, so A will come into here and join up over here. We have a particular, for example, another A gonna come in here again. The DNA polymerase is gonna join it up together. So DNA polymerase is gonna run along the parent strand to add new nucleotides to the parent strands. Okay, so nucleotides join up following the base pairing rule. Again, it's ATCG. Now, the cool thing is, addition of new nucleotide always starts from the 3 ends of the parent strand. What does that mean? So it always starts from the 3 ends. So for example, in this case, DNA polymerase is running upwards. Okay, so we're just gonna draw. We'll use a black pen here. So DNA polymerase is running this position, okay? Now, if it's going up this position, that means DNA polymerase for the lagging strands will be running down from this end, okay? So going down here and going down this way, okay? So this is step 2. What happened is the separated parent strands are paired up with new nucleotides. So we get these new nucleotides from foods or nutrients with the help of the polymerase called DNA polymerase. Okay, this is another enzyme that we talked about. First one is the helicase. It unzip or unwinds, unzip the parent DNA, and DNA polymerase add additions of new nucleotides to the existing parent strand. It will always add it from the 3 ends of the parent strand. So 3 ends, for example, the leading strand is going upward, the other one will be going downwards. Okay, so this is step number 2. Okay guys, we are going through the last step of DNA replication. Okay, so DNA replication for the leading strand is continuous. I haven't drawn the helicase or the DNA polymerase because if I do draw it, they'll be really messy. Okay, so in your exam, what you should do is you should have multiple drawings for each step of your DNA replication, if the question is asking you to explain DNA replication in detail. Now, I am gonna pretend I'm going to pretend this pen, what I'm drawing here, is the DNA polymerase, okay? So imagine you are the DNA polymerase. What you're doing is you are joining the new nucleotides to the original DNA strands. Now, for the leading strand, it is very easy for us to join up because we can read it, in a very perfect sense by facing this way. So everything is facing us. We can, read it really clearly, so we know what nucleotide needs to be added, and that is why DNA replication for leading strand is continuous. Okay, so we're going to start from the 3 carbon, or the 3 locations of this leading strands. We can see it's T, so what it does is that we just basically grab adenine nucleotides from our food or nutrient, and we just write A here. Okay, again, similarly A, G, C, A, C, G, A, and just gonna leave it there. Okay, let me just add over this one here, T. So as you can see, for leading strand, it is really easy to add up because we can read it in a very cool way, and it's a very smooth way to read it. We don't have to do anything with it, so we just read it, and so the replication process is continuous. However, the DNA replication for the lagging strand is discontinuous. Imagine you are the DNA polymerase. You can read a leading strand really easily. However, for the lagging strand, it is other way around. It is really hard for you to read. So what do? It takes you a much longer time to replicate, and you need the help of something else as well. That's what we're going to talk about. So first of all, we're just going to try to add it up. So this looks like a T. So again, imagine I'm the DNA polymerase. I'm going to add an A here. Okay. Now, I'm kind of confused with this one. I'm not entirely sure what it is. Some of you guys might know it is an A, but I'm just going to leave it there. And I'm going to read next one. I go, oh, that's a C. So I'm going to put a C here. And okay, next one is probably a G. And again, I don't really know about this one. I don't really know about that one either. I'm going to continue down. C and T. T. Okay? Now, the newly synthesized nucleotide fragments. So these are called the nucleotide fragments. Okay? So I'm just going to go. Nucleotide fragment. Now, these fragments, the ones that I know of, are called the Okazaki fragment. Okay, Okazaki is this Japanese guy. He discovered this amazing little process, and he actually won a Nobel Prize for it. So, because the DNA polymerase cannot read the sequence properly because this is facing the other way, what happened is that we form these little fragments called nucleotide fragments. Okay, now Okazaki fragments can be joined up together with the help of the third enzyme in this whole process. It's called the DNA ligase. Okay, so I'm going to use blue. So DNA ligase is much smarter than me, or imagine it's my older sister. She's always smarter. She came along and say, hey, look, I know how to join you guys up. So there's an A, this one is a T, and this one is a T as well, and this one is a G, and that's it. So the DNA ligase join the nucleotide fragments together. So eventually, on the lagging strands, you're gonna get a sequential. You're gonna get a new strand of DNA. Okay, the nucleotide will join up as well, just like the leading strand, but it takes one extra step with the help of DNA ligase. So at the end, you're going to get 2 identical DNA strands, okay? Again, repeating this, the DNA replication, or this replication fork, occurs multiple locations along the DNA strands, okay? Not just one. It doesn't just start from here and just go all the way along and eventually stops. It actually opens up multiple places, and eventually the DNA will be replicated, okay? Leading strands is really easy to read because it's the sequence is reading down like this way. It's really easy for the DNA polymerase to read and just add it up. So leading strand replication is always continuous. DNA replication for the lagging strand, however, is discontinuous because all the letters are facing the other way. The base are facing the other way. It is really hard for me, the DNA polymerase, to read. So I form these little islands of nucleotides or fragments called Okazaki fragments, okay? Now, Okazaki fragments are joined up, or with the help of DNA ligase, which is another enzyme, by joining them up to form a line of new strands of DNA. And at the end of DNA replication, you will get 2 identical DNA strands. All right, summary for DNA replication. So DNA strands are always anti-parallel. One strand is going downwards, the other strand is going upward, okay? So it's always anti-parallel. Now, the leading strand is always from the 5 to 3 direction. In your exam, you can just say from the 5-3- direction. Don't really have to talk about the carbon position because that is university level, okay? You just have to know that DNA strands are anti-parallel. If you don't remember the leading and lagging strand, that is fine. Just remember it is always anti-parallel. Now, the enzymes involved in DNA replication, 3, okay? Helicase, unwind, and unzip. I forgot to write something. It also breaks the DNA, breaks the H bond. Break H bond between base, okay? So, helicase is super duper important. It does 3 things. It first of all unwinds the double helix, unzip like a zipper, and also when it unzip, it breaks the hydrogen bond that is between the bases, okay? So actually bases, excuse me. DNA polymerase joins the new nucleotides with the parent strand. So you will now have 2 strands of DNA, okay? One is leading, one is lagging, depending on where the cap is. And the DNA polymerase is going to come along and help join the new nucleotides with the old parent strands, okay? Eventually, you will get something like this, okay? Just like with my pen, all right? So like that. Now, DNA ligase join Okazaki fragment in lagging strand because, again, just like the previous video, well, not video, but the previous board, the leading strand is really easy to read and therefore the replication is continuous. However, in the lagging strands, because it's so hard to read, it needs the help of DNA ligase to join up the little fragments of nucleotides, alright? So DNA ligase join Okazaki fragments in lagging strands. The really important enzymes, probably the helicase and DNA polymerase. DNA ligase, you sort of have to talk about it when you start mentioning about leading and lagging strands, okay? Now, the original strands of DNA act as a template. So, our original strands of DNA, for example, this is my original strands. One is leading, one is lagging. When you separate them, they act as a little mapping tool or a template where new nucleotide can join them. This just to ensure that there's nothing wrong with DNA replication, okay? If something's wrong with the DNA replication, normally we have mechanisms to correct that, but sometimes when those mechanisms disappear or disabled due to mutation, that's when cancers occur, okay? So you're gonna get a random newly synthesized DNA strand. That might not work as what we wanted to do. So, original strands of DNA always act as template. Lastly, DNA replication is semi-conservative. That means the newly synthesized DNA strands will have half old, half new. Same thing over here, we have the half old and half new. So it is semi-conservative, okay? So conservative just means old, but because there's a newly synthesized DNA strands with each of them, it's called semi, so half. Half old, half new. I hope you guys find this video helpful. What I'll do is I will also put this in the Word document so you guys can look at the Word document while going through this particular video. Hope you guys enjoyed this, and I will see you guys next time.